Thank you very much for the opportunity to talk to you. For those of you who don't know Oxtec, we're a small UK-based company funded entirely by private individuals, family offices and grant funding. We're working on a new approach to combat vectors that spread disease, and I'm going to change both continent and disease from the last two speakers. So I'm going to move from Africa and malaria, and I'm going to come over into the Caribbean. So chikungunya, you are probably familiar with, but for those who aren't, I mean, this is a mosquito-borne disease for which there's no medication and there's no vaccine. It's spread by Aedes aegypti. Chikungunya literally means to be bent over because you get intense joint pain. Most people should recover quite naturally. What seems to be a feature of this disease, though, this virus, is ongoing complications. It seems to have a great ability to exacerbate existing ailments. And in a recent conference in the Caribbean Health Authority, Public Health Agency, was saying to up to 50% of the people who'd had chikungunya were still reporting symptoms well after 6 months. Now, that's a higher figure than I've seen elsewhere, but it will tie up health resources, cause a lot of pain individually and a lot of state expense. So chikungunya came into, normally we associate it with Africa and India. It came into the Caribbean really for the first time in December 2013. First case in St. Martin. And then by the end of that month, it had spread to another 2 or 3 islands. And then within about 6 months, it had spread throughout the whole of the Caribbean. And then if you take a count now, there's now about 1.2 million cases of chikungunya suspected around the Americas region, a quite astonishing rate of growth. We shouldn't really be astonished because the vector, Aedes aegypti, is very well known. If you take dengue, which it's often associated with, dengue was reintroduced into Brazil, for example, in the early 80s, and now there's over 1 million cases every year. So let's look at our enemy, and here she is, taking a blood meal. Aegypti, as you'll probably know, highly anthropophilic, and with a short flight range, which basically, take those two things together, means that Aegypti needs to live in and around your home. That you are the food source, we are the food source, or the blood meal. So it needs to live in and around your home, which means if you're going to try and tackle Aegypti with conventional tools, mainly insecticides, you've got to get into every single house on a regular basis, which in an urban environment, a sophisticated urban environment, is really very difficult indeed, if not completely impossible. So our solution is rather different to using chemistry, can be used in combination actually, but we have genetically engineered Aedes aegypti, and we've engineered it to really produce an effect that's a follow-on from the sterile insect technique that's been used in agriculture for many, many years, using radiation as the mechanism of sterilization. But here we're using genetics, and so we're sending out males, which when they mate with wild females, the offspring die. And that's it. It's a relatively simple concept. We send out the males, they mate with the females, the offspring die. We send out more males, they mate with more females, the offspring die. And if you think of an Aegypti female laying up to 100 eggs at a time, if you're taking out that reproductive capacity, you crash the population really very fast indeed. So the genetic engineering here, there are 2 principal genes, and both, I would suggest, are very important. Now, the very first one is a self-limiting gene, and what we mean by that is it actually blocks the reproductive capacity, so it will kill the offspring. So our male will mate with the female, the offspring will die before they become reproductive adults. And actually, we don't do this to every single insect. We actually created a strain of Aedes aegypti. It's called OX513A, which shows we're rather better at science than marketing. But OX513 was actually created as a strain in 2002. And since 2002 to about 2009, we were doing internal trials, contained trials, checking the product worked, and then we've actually gone into the outdoor world from about 2009 onwards. So years of development. So the very first gene we've introduced is a gene that kills the offspring. It's self-limiting because it will die out in the environment because the insects can't reproduce. The second gene there is a fluorescent protein, which means that actually you can detect the insects. You can detect the offspring, which are going to die, because when you use a light with a special filter, they will show up as red, which means you can actually moderate the releases as you're running through a program. You can detect where they are. You can be very, very precise. So our whole process is one built on metrics and monitoring, in fact. It's a very, very precise, organized system. So those two aspects, a self-limiting gene and a fluorescent protein. Now, we obviously don't want to keep genetically engineering mosquitoes. That would be too expensive. So we have a very simple system of turning off the self-limiting gene in a factory, which is that we actually rear the mosquitoes with an antidote, which means that the lethality doesn't kick in. So actually you can produce millions and millions and millions of insects in a laboratory, very simple, keep reproducing them at very low cost. But when you go out into the outside world, they don't have access to the antidote, and that self-limiting gene causes the death of the offspring. So just conceptually then, it is a simple process. You take a town, you decide where you're going to release, and you send a truck down the street, and literally there is a man in a van, it could be a woman in a van, and they will release the mosquitoes as they go. And we release them out of little pots, about 1,000 at a time, and the flight distance, again, of Aedes is about 100 meters or up to 200 meters in total. So actually, as long as you plan your program so that you're getting spatial coverage of the area that you want to release in, you will actually control the population of Aedes in that area. And equally, you won't control the Aedes in areas where you don't release, which from my point of view is quite a good thing as well, because it's all about control. And here is the van. You can see it's pretty unmodified, apart from some rather fancy artwork with Aedes transgenico on the front, and a rather neat loudspeaker that sings a jolly song as it's going along, explaining what it's doing. It's a very annoying song, I have to say, but it's very effective. Okay, so the benefits of this approach, well, first and foremost, efficacy. You're using the male to go and seek a female, and males are very good at seeking females. Male mosquitoes can seek females much better than a fogging truck or a man with a chemical backpack. We're releasing males because males don't bite, so males are fairly innocuous. You might think that would apply to males of different species, but with mosquitoes, they're fairly innocuous. So you're releasing the male to mate with the female. It's very much a species-specific approach, so you're really actually affecting the one target insect in the area that you wish to, and you're leaving other insect life untouched. The protein that is produced that actually causes the death of the offspring is not toxic, it's not allergenic. If you're bitten by a genetically engineered mosquito, as I am pretty much every week, then there is no difference to that, to being bitten by a normal mosquito. And then you have this heritable marker, so you can actually see what you're doing in the environment. And possibly the most important aspect here is it is self-limiting in the environment. This is a biological control mechanism, and with any biological control mechanisms, you really want to be in control, you want to know what you're doing. And this is self-limiting. It doesn't perpetuate or spread. It's the exact opposite to the perception people have of using genetic engineering. If you mention GM crops, genetic engineering, people always have this fear of the genie coming out of the bottle. You're putting in motion things that can't be undone. And this is actually the opposite. It's a self-limiting system. It's a dead end. So here are the field results that we've done so far, starting in 2009 across. So in every single field result we've had, and so by what I mean by a field result, I mean take a town, a neighbourhood, anything up to about 5,000 people living in an area, take nearby towns which are very, very similar, treat one, use the others as untreated control sites, and look at the effect you're having on the Aedes aegypti population over a period of about 6 months. So in every single trial, we've got over 90% control, and this is irrespective of whether you're releasing in the rainy season, the dry season, or moving from the wet to the dry. So a really fairly good result. Now, when we try and compare this to the use of chemicals, and if anyone's got data, I'd be grateful for it. We can find studies where people have controlled an urban population of Aegypti in a similar sort of environment to about 30%, and this is 90. The Florida Keys Mosquito Control District, who we're working with in the Keys, obviously, in Florida, they say the best they can achieve with all of their system is to reduce the adult population by 50%. So again, this would look to be fairly favourable by comparison. All our trials actually have been done independently. We've obviously been involved closely, but they've all been led by external evaluators. The one that will take place hopefully in the Keys shortly will actually be the first one that we've done in our own name, and that's an outcome of working with the Food and Drug Administration, where we have to be the lead. So it's all very well getting that population under control. How do you actually keep it there? And how do you keep it there in a very cost-effective way? And I think there are numerous options here. The first one probably is the least attractive, I think, which is that if you imagine that green blob as a town, and you imagine that town, we've pretty much controlled the Aedes population down to very, very low levels, but it's surrounded by other towns who haven't used the same approach. So how would you keep that area clean from reinfestation? Well, the first thing you could do is just carry out a continual low-level release using a very small number of males. So as females come back into the area, brought in by freight or transport, then the males will mate with them and you'll keep it under control. You could also look at that approach and say, well, I'll just do this seasonally. I'll do it once a year. I'll just take out that seasonal peak in mosquito numbers. So that's a variation on a theme. What you also might want to do is, particularly if you're a Caribbean island and you're surrounded by water, is actually look for island-wide elimination, actually eliminate Aedes off the island. It shouldn't be there. It should only be in North Africa, so you're getting rid of an invasive species. If you achieve that, then actually the strategy would be look at your ports of entry and carry out a border program, if you like. Release around your airport, release around your marinas and ports, because that's where they'll come back in. Another option you might want to do is treat hotspots. Really fabulous presentation on Madeira earlier today with a very clear delineation of the hotspots in Funchal where the major risk was being represented by Aedes. Well, you could actually just target those areas and basically take Aedes out. The other solution, which I think is also quite a good one, is do nothing at all. Just monitor. You've got to monitor, but use our process as a retreatment if you monitor and Aedes comes back. Another inflection here is using insecticides. If you could use insecticides in combination with our program, or you could use our program to reduce the population and then try and maintain it with sprays. And if that doesn't work, come back to us. I think a lot of people will dream up different scenarios. There's no one right answer. A lot of people actually ask me, what's the cost of this? And actually, it's a very, very difficult question to answer. It really depends on what your objective is. The cost of the top left, an area-wide continual release at low level, is very, very different from the one at the bottom here, where the cost could be virtually near zero. Equally, the cost of actually taking out the seasonal peak in mosquito numbers is very different from trying to drive it down to below 90% and keeping it at trace levels. So it really depends on your objectives and how you look to integrate this approach with others. The other thing I'd bear in mind is that when we look at our costs, we're going through a fantastic learning experience. developing something really new like this, you can't really go to anyone and say, how do I produce tens of millions of ADs at scale really cheaply? Nobody knows. Lots of people produce ADs, but they do it in laboratories. So you have to work it out yourself. And a couple of years ago, we were really proud of ourselves because in quite a small room, we could produce about 2 million a week. In that same room with the same number of people, we can now do about 25 million. And there are just numerous ideas within the team of how to improve various different aspects of rearing, of handling, management, release. We've got far more ideas than we have money to deploy and far more ideas than we have people to carry it out. But I think what we'll see is a very, very sharp changing cost profile over the next few years. And actually, at the moment, I can't really give you a precise figure of where we're going to end up because I just don't know. What I do know is the more we work on it, the better it gets. So those are some just snapshots of sort of theory of how you would maintain control. In terms of regulatory progress around the world, we're actually doing this in agriculture as well as with mosquitoes. So this is a composite chart of where we have regulatory approval. Now, the US actually did the most comprehensive environmental study on our technology. Actually, with GM crops, you do something called an environmental assessment. With us, they went for an environmental impact statement, which is a much more heavy document and study. And they came out with the conclusion that our approach was environmentally preferable to other forms of control, which is obviously very nice of them to say that. But from an environmental point of view, it's pretty good. It's targeted, it doesn't hang around, you can tell where it goes. But we've got various different approvals. Possibly the most exciting, obviously the right-hand side where we've done outdoor release or about to do it. And then in Brazil, we now have what we call technical approval for commercial release, meaning we've passed through all safety and environmental hurdles in Brazil. So we can go anywhere in Brazil now and release our OX513A mosquito. And hopefully that trend will follow in other countries. Final slide for me then, and really it's a thought piece that says, when you're introducing a new technology, whether it's something in common with what's been done before and a variation on a theme, you always have to go through a regulatory process, and you're trying to balance efficacy with safety and environmental issues, and that's absolutely critical. I think, though, that there's an extra burden on new technologies, new paradigms, particularly when you're in the biological space. Because we have some pretty, as humans, we have not a very good track record sometimes on introducing new technologies. And in conjunction with efficacy and safety environment, I'd like to introduce this concept of control. Well, I would like to. There we go, control. And by control, I don't mean, in this instance, controlling the population, because that's already sort of covered in efficacy. I mean being in control of what you do. Absolutely fundamental. If our truck goes around the town releasing mosquitoes, how do we know whether the truck driver has actually covered all of the town? How do we know whether we're releasing the right number of mosquitoes in one part of town versus another? Because we know the incidence will be different in terms of different parts of town. How do we know that our mosquitoes only travel 200 yards? How do we know all these things? And how do we answer all the what-ifs? Now, we can answer these. We've got a lot of studies. But the other really crucial question is monitoring. And we have a built-in track and trace. Every single mosquito, every single fruit fly, everything we have is actually tagged. It's marked with a colour. So you can tell, basically. You can compare. You can look in the field, and you can see where our mosquitoes have gone. You can see the effect they're having. You can moderate the release rate. So you are actually in control of this program. That's what we're doing. Thank you very much indeed.